I'm thrilled to see you all here. I'm Lucy Lipovsky, the chair of the program committee, and it's wonderful to have you. And tonight we have a phenomenal guest, Fiona Crawford, who's one of our members, one of our new members. And she is the president and the CEO of the Roskamp Institute, which she co-founded and which became a standalone research institute in 2003, and is now home to a staff of more than 60 scientists, clinicians, grad students, and research assistants. She moved to Sarasota in 1992. When you hear her talk, you'll know she isn't from here. She has a bachelor's degree in biochemistry and genetics, which Karen Holbrook also is a scientist, and maybe some others on the call, and from Queen's University in Belfast, Northern Ireland, and a PhD in medical genetics from the University of London. Fiona not only runs the Roskamp Institute, which not only deals with Alzheimer's, but traumatic brain injury, post-traumatic stress disorder, Gulf War illnesses, and she told me she just got a grant and they're working on the relationship of, I guess, health, but she can tell you more, and red tide, which I'm not in Florida now, but I keep reading about, but my colleagues and friends there are probably understanding and fearing it more. So besides just running the Roskamp, she also, I think if I get this right, has a nonprofit, and she's the chief operating officer of Archer Pharmaceuticals and a spin-off for-profit research organization. So she does lots of things, has written more than 175 papers, and we are incredibly fortunate that she's going to share her knowledge with us. She's going to talk to us for about 30-40 minutes and then answer all of our questions, although I promised her we would not stay on all night. So with that, Fiona, go for it. And thank you so much for agreeing to do this.
Thank you. Thank you so much. And thank you for the invitation. And let's see if a year and a half of using this technology has actually showed that I can master anything.
Let me suggest that everyone mute mute themselves so that we don't have background noise.
Everybody see the screen?
Yes.
Yes.
I'm hoping everybody's okay. Good. All right. Thank you. Thank you again for the invitation. It's a pleasure to have this opportunity to talk to you all. As I'm sure Karen, who's spoken to me at length about some of what we do, and Lucy already knows, I could probably talk for hours and hours. I will endeavour not to do that. So I've got some high points I want to hit, which there's so much could be said. I'm going to try. I'm not going to dive into the molecular biology that we do at the Institute. I'm really going to focus on things that I think will capture some of the top line areas of interest for you.
Seen one get up here.
But I figured that if I sort of present a bit of a smorgasbord, then when we get to the questions, anything you are interested in, we can take a deeper dive. So just getting, can you all hear me okay? Mm-hmm. Okay, great. So as Lucy already said, the Institute is a not-for-profit right here in Sarasota. We have many different disciplines under our roof, and we also have our clinic, which a lot of folks are familiar with. It's a neurological clinic focusing on Alzheimer's disease, Parkinson's, multiple sclerosis. We also work with traumatic brain injury patients, PTSD, and an awful lot of our clinic patients also participate in our research studies and our clinical trials. And it's a fantastic juxtaposition because for the scientists, it means that every day in our clinic, they're seeing the patients coming and going, patients who we are trying to treat. It's the daily reminder of our battle, what it is we're up against. Because we spend all our time in the labs dealing with the mice, dealing with the test tubes and the petri dishes, but they're just in one corner of our building are the patients and their families. And it's a great reminder. And it also means we have that juxtaposition all the time of what's clinically relevant. As scientists, it's, science is just fascinating and exciting and always new. And we can chase off down all sorts of avenues. But we need to make sure what we're doing at the Institute anyway is all entirely focused on an end game in the clinic, better treatments, better diagnostics for the conditions we're working on. So we're careful to continually review what we do with a clinical context and make sure that we're still pursuing a path that will take us to the clinic. So our institute, generally our approach is to use or develop animal models of the conditions we're working on. And that, of course, is because in animal models, we are able to chart the course of Alzheimer's disease or whatever condition it is we're working on from the start all the way through. And a mouse has typically a 2 to 3 year lifespan, so we can actually explore the lifelong consequences in a reasonable time frame that, maybe a reasonable time frame that can be funded by the National Institutes of Health, for instance. So a lot of our studies are in mice, looking at behavior, looking at pathology, looking at what changes, what's the difference between a mouse that has Alzheimer's disease and a mouse that doesn't? What are the molecular pathways that are altered early on? How do they change with time, with progression of the disease? And the idea is that by dissecting those molecular pathways, we will get to identify new targets for therapeutic intervention. And I'm describing all of this. Firstly, of course, I'm enormously proud of the Institute, but this is a bigger subject. This is about Alzheimer's disease and where we globally are at with Alzheimer's disease. But the Institute operates very much like a small biotech company. So what we are doing as regards Alzheimer's disease is what is being recreated on a much larger scale by the pharmaceutical companies. So we have our preclinical models, we start off in cell models, then we move to animal models, and then we make that big translational jump into humans if possible. We see if we can jump across that gap and have something that really has efficacy in the human population. We have a lot of exciting tools and toys now that we can bring to bear in these explorations. We have behavioral testing for the animals. We can look with our mass spectrometry instruments. We can get down into an amazing level of detail of detection and analysis of proteins, lipids, metabolites in the brain, in the blood. And then, of course, we have neuropathological analyses, just like we do for autopsy tissue for human cases, where we can look at sections of the brain and not just see what's changed anatomically, but also whether we see changes in proteins and molecules that we think are relevant, and whether or not any difference has been brought about by any treatments we may have tried. So, as I said, this talk, I'm going to cover a lot of different things, so it might jump around a bit, but I'll be happy to go into more detail on anything that captures anybody's attention particularly. One question that always comes up is, what's the difference between Alzheimer's and dementia? And it's a very simple answer. Dementia is the umbrella term for conditions that lead to cognitive impairment, loss of intellectual ability, that is brought about by neurodegeneration, loss of or damage to neurons in the brain. Alzheimer's specifically is the most common form of dementia, which is, as unfortunately we're all probably very well aware by now, it's characterized primarily and early on by short-term memory loss and then progressive cognitive deterioration. So what exactly is Alzheimer's, and why is it characterized by memory loss? So in the human brain, rough estimate, we've got 86 billion neurons. So, I mean, that's just a mind-boggling number, but if we think about the complexity of the brain and the speed at which thoughts occur and ideas, and we can get an idea of how dense the neural network must be, and the number of connections between one neuron and another. There are thousands of connections, and the whole network is connected throughout the brain. The neurons are connected by synapses, and the more densely connected the brain is, this usually does seem to correlate with levels of cognitive ability. And certainly as Alzheimer's progresses and we see a progressive loss of neurons, we see a loss of synapse function. So the point at which one neuron is speaking to another, if you will, is lost. And what happens is that signals are sent from one neuron to the next, to the next, to the next in fractions of seconds. And the messages get spread very quickly throughout the whole brain, and then the instructions are sent wherever they need to go in the rest of the body for how to respond to these cues that are coming in from the environment. Now, the first place really that we see pathology in an Alzheimer patient is in the hippocampus. which is this part of, right in the middle of the brain. Hippocampus is actually the Greek for seahorse, and it was so named because this part of the brain anatomically looks, to some people, I guess, a little bit like a seahorse. But it's right there in the middle, and new information comes from our senses into the hippocampus, and then it's processed in the hippocampus, and then it goes to the cortex for a process called consolidation, which is where the long-term storage happens. So if you think about that, and if you think that the hippocampus is the first place where we see problems and pathology in Alzheimer's disease, it starts to make sense why it is that somebody with Alzheimer's disease might be able to remember what they did in the summer of 1950, but they don't remember that you already visited them that morning and had breakfast with them. It's because that new information, it arrived at the hippocampus, but it didn't get processed properly. And so it never went to that next stage of consolidation and really storing it. So those short-term memories are not retained. And a lot of folks, you know, they talk about their relatives or friends with Alzheimer's disease, and they'll give the exact example I just said, you know, oh, but his memory's great, you know, he remembers things from such a long time back. And that is very typical, but they will not remember what just happened. It's a, you know, there's no need to say it, because I'd be surprised if there's anybody, certainly on this call, who has not experienced Alzheimer's disease through a friend or a family member at this point. It's a horrible, horrible disease. It affects not just the patient, but of course the entire family, and they suffer too, along with the patient. There's lots of issues associated. We can talk about those afterwards, but there are certainly issues of caregiver stress, the folks who are looking after Alzheimer patients, and of course there's a lot of guilt involved in family members when they finally make the decision for their family member to go into a care facility where folks are trained to look after people with dementia and people with Alzheimer's disease, but that can bring a lot of problems for the caregiver. So a really, truly devastating disease. And, you know, I think a lot of people would certainly, if they had a choice, would go another way rather than Alzheimer's disease. So this little cartoon is just to talk about the two main pathologies of Alzheimer's disease, which were identified by Alois Alzheimer back in 1907. On the left, we see the normal brain. There's the neurons. And on the right, we have these two entities, amyloid plaques, which, as you can see there, they are outside of the cell. These are clumps of a protein called amyloid. And then neurofibrillary tangles, which are inside the neurons. You can see there's a sort of a bit of a tangle shape inside the neurons there. These two structures were identified and have to be seen at autopsy in order for a person who has died to be pathologically diagnosed with Alzheimer's disease. These are both caused by normal proteins. Amyloid is a normal protein. Neurofibrillary tangles are comprised of tau, which is a normal protein. We all make these proteins all the time. They have a normal function. But this is an example of what happens in nature. There's not really black or white with anything in nature. You don't have good proteins and bad proteins. You have proteins that do things in one environment and do something different in another. And when you have a changing landscape and a changing environment or changing messages coming through, then things can change and we can get these detrimental effects. The other pathology that is widely recognized in Alzheimer's disease, but also in many other conditions as well, is neuroinflammation. So inflammation in the brain is massively important in Alzheimer's disease. As these pathologies, particularly the amyloid plaques, as they develop, they create a lot of neuroinflammation in the surrounding area. The brain of an Alzheimer's patient is highly inflamed, just like the brain of a patient with traumatic brain injury or patients with many other conditions. And inflammation is one of the key things that we are incorporating in our analyses now, because these things all work together. This isn't a situation, nothing in nature really is a situation where something is operating in a vacuum. We need to consider the whole brain together. And I mentioned the mouse studies earlier. I do always like to mention that if we could do what we need to do, just looking at cells in a petri dish, we would, but we do not have the technology at this time. to mimic the complexity of the human brain in a petri dish. We have to look at living animals. And so we have to look, ideally, as I said, we use mice, but we have to be able to look at how everything in the brain works together. The neurons, the blood vessels, the inflammatory cells, the microglia, the astrocytes, all of the different components of the brain, how they all work together. Because if we just look at one piece, we'll only get one piece of the story, only one piece of the jigsaw puzzle. So there's a real picture. This is an actual section from the brain of somebody with Alzheimer's disease. And you can see here in black, I don't know if you can see my arrow there, but that's a neuron with neurofibrillary tangles inside. And these stained pink, these are amyloid deposits, these clumps of protein. And amyloid and tau, as I said, they're normal proteins. Amyloid, we still don't really know what amyloid does for a living. A lot of our work has suggested that it has a role in blood vessels, maintaining and growing new blood vessels. But when it starts to build up, what happens is it stops being a soluble protein anymore, and it comes out of solution and starts to build up. I always use the analogy of adding sugar to a glass of water. At some point, it stops dissolving, just sedimenting in the bottom. And amyloid building up in the brain is very sticky, and it starts to stick together, and it forms these clumps that you can see. And then that starts to create a neuroinflammatory reaction around it. And similarly with tau, tau is a structural protein. It's inside the neurons. I like to think of the neurons like highways, where you're traveling down one neuron to get to where it connects at the synapse with the next neuron. And this is how the messages are getting transmitted. And I usually use the example of the I-75 and trying to get transport from Tampa to Naples, let's say. And if everything's running smoothly, that's great, but when does that ever happen on the I-75? So, maybe somewhere around University Parkway area, there'll be an accident and there'll be a lane that's closed down, and then everything starts to slow down. So the idea there, the analogy is, if tau starts to break down the infrastructure in the neurons, and the transportation can no longer happen along the length of a neuron at the speed it's meant to, and maybe there's a block and the other lanes have slowed down, there might be a complete block if the accident was really bad, if there was something that happened right across all the lanes of the interstate. And so the information can no longer get through. So it doesn't make it to the end of the neuron and doesn't get passed on to the next neuron. So that's how the signals start to not get transmitted from one neuron to the next. So tau, when it stops being that scaffolding protein that's holding everything together, also when it detaches, it too sticks to itself and starts to form these aggregates. And that's how these neurofibrillary tangles are formed. Now, we have the ability these days to image both tau and amyloid in the brains of living individuals. And this is an interesting representation here, because the images on the bottom are of different individuals and looking at amyloid, and at the top, the same individuals, and we're imaging for tau. And the amount of yellow, green, red is indicating how much of that protein is building up in the brain. So the two on the left, you can see this top image, there's hardly any tau, just a little bit here, and there's a fair bit of amyloid. This is a normal individual, low amyloid, low tau levels, cognitively normal. Very interestingly, the person in the middle has an awful lot of amyloid, the whole brain just full of amyloid there. Not too much tau, but this is also a cognitively normal individual. It's only when we go to the person on the right who has quite a lot of tau, and you can see particularly here in red, some very concentrated areas of tau pathology. This individual, who also has all of this amyloid, they have the cognitive problems. That's the AD patient. So this really shows us that it's the tau pathology rather than the amyloid pathology that correlates best with cognitive function. And this is something that actually has only been revealed in the last 10 or 15 years. And interestingly, in a lot of the clinical trials that have taken place, which, as I'm sure you know, have by and large been unsuccessful, it has been shown that the treatments in many cases have been able to clear amyloid out of the brain, but the person has not shown any improvement in cognitive function. So essentially, removal of amyloid from the brain doesn't do it. And really the reason that is because of the timing and the progression of Alzheimer's disease. So I think you can hear me okay, because I'm hearing some feedback. This graph, this is from a publication a few years back, which attempted to sort of map the different biological changes, the biomarkers that we see during the course of Alzheimer's disease with the actual disease stages. So just to get our bearings here, along the x-axis, we have time. And on the y-axis, we have the amount of each of the biomarkers under consideration. So the time axis along the bottom, everything up to here is considered a cognitively normal individual. And then this little bracket here is individuals with mild cognitive impairment. And then beyond that, it's individuals with full-blown dementia. But if we look, all the while people are still considered clinically normal, we're seeing increases in the amount of amyloid in the cerebrospinal fluid, the amount of amyloid being detected in the parenchyma, in the brain tissue, the amount of tau being detected in the cerebrospinal fluid, the changes on PET imaging for glucose, showing us blood flow. All of these are increasing significantly long before somebody reaches the point where they get a clinical diagnosis, where they're diagnosed with mild cognitive impairment. So by the time somebody reaches that stage, and even further on when they actually get diagnosed with dementia, all of this has been going on for a long time, 10, maybe 20 years. And so this is one of the problems with what we're dealing with in trying to identify treatments for Alzheimer's disease, because we're looking at what we see in an Alzheimer brain right now, or what we see in an Alzheimer brain at autopsy. But what we're looking at are the consequences of many, many years of disease. So how do we get back? I mean, it seems intuitive that the earlier you can tackle a disease, the earlier in the stages of the disease that you can tackle it, the more likely you are to have better effects. But this could be something that's going on for 20 years before we actually have sufficient pathology for it to present a clinical problem. So this is a real issue. And so much of the work has been focused on amyloid treatments to try and reduce the amount of amyloid in the brain. And I've just told you, a lot of those treatments have shown the brain doesn't change anything but the cognitive performance of those patients. And that's because it may all start with amyloid, or amyloid may be one of the initiating factors, but very quickly, all these other things start to come into play. And that includes inflammation. And we know that inflammation contributes greatly. And you can then take away the original source, the amyloid that was there right at the start that caused the problems. But now you've got widespread inflammation in the brain. Taking away the amyloid, it's too late. You've got all these problems. You have to tackle multiple things together. It's not going to be enough to just tackle one part of the problem. So there are a lot of modifiers of risk of Alzheimer's disease. I will talk in a moment about some of the very specific genetic risk factors for Alzheimer's, the genetic causes of Alzheimer's, but they are very rare. So for most of us, these are the key things that can enact our risk for Alzheimer's disease. So you may have heard of ApoE, apolipoprotein E. This is a lipoprotein. It carries lipids around the body, and it exists in three common forms, E2, E3, and E4, and it's been known for a very long time. It's associated with atherosclerosis, and about, more than 20 years ago now, it was identified to be also a risk factor for Alzheimer's disease. The E4 form of ApoE is a risk factor. Now, I say that with caution because folks tend to jump on these ideas of genetic risk factors, but it doesn't mean you will necessarily get Alzheimer's disease if you are an ApoE4 individual, but you have increased risk, and you would also get the disease earlier than somebody with only E2 or E3 forms of ApoE. We still don't know exactly how ApoE contributes its risk for Alzheimer's disease. And in fact, it's an interesting protein. I think it's one of the most interesting proteins I work on because it confers risk for multiple situations, multiple disease conditions, including traumatic brain injury. So it seems to me that ApoE is more about risk for outcome after injury to neurons, such that if you carry the E4 form, you are less able to recover after an insult or an injury versus the other forms. So that's the biggest known risk factor for Alzheimer's disease outside of the disease-causing mutations. The other factors that I've listed here, I won't go into all of them in a lot of detail, but we have known for some time now that if you protect yourself against vascular disease, that is also going to protect you against Alzheimer's disease. So controlling your hypertension, controlling your diabetes, anything that can improve your vascular health is also going to improve your chances of not getting Alzheimer's disease. Head injury, we've known for a very long time, is a risk factor for later life neurodegeneration. It's kind of been reinvented in the last 10 years because we've suddenly realized that getting hit on the head every Sunday is not a good idea, but it's been known for a long time. The interesting thing about head injury, which of course I run a whole separate program on traumatic brain injury. The interesting thing is that it doesn't just contribute risk for Alzheimer's, it also contributes risk for ALS, Lou Gehrig's disease, or for Parkinson's, or for Lewy body dementia. So there's some interesting additional factors at play there. So really TBI is a risk factor for later life neurodegeneration. And it seems that there are additional factors that then might influence what particular presentation that might take. We know that our immune systems and inflammation play a role, hormonal balance, stress, toxic exposures. Sex differences, that's a confusing one. It used to be very widely thought that women were at increased risk for Alzheimer's disease. Then it was decided that no, that was actually a distortion of the data, and the perception of women, there being more women with Alzheimer's, was all about the fact that women live longer than men. Now we're swinging back again because it turns out that the differences cannot just be explained by the fact that women live longer than men. There are additional features. A lot of these are just to do with the basic sex differences. There are things going on in women, such as hormonal changes with age, that influence outcomes and influence Alzheimer's pathology that men don't have. But there's also things that influence men. So, for example, women are more prone to depression, which can have a negative effect on your risk for Alzheimer's. Men are more prone to head injuries. That's having a negative effect. So, despite the fact that it seems like such an obvious question to ask, we still haven't got to the bottom of the molecular basis of any sex differences in risk for Alzheimer's disease. Sleep, I'd like to mention, because I think it's a fascinating area, and we're just starting a program on that at the Institute. It's been relatively recently identified that we have a system called the glymphatic system in the brain, which essentially acts like the trash collection. And what happens is that during the course of the day, there's debris created all the time. You know, cells die, there's things get in there, and they're cleared out. They're cleared from the brain tissue into the interstitial fluid, into the cerebral spinal fluid, and then cleared out of the brain. And that would include little bits of amyloid, little bits of tau that are around. They should get cleared out in that system. So if that system isn't working well, they don't get cleared. The trash doesn't get picked up, in essence. And so the trash starts to build up, and then it starts to form those pathological features I showed you earlier. When does the glymphatic system do its best work? When we're asleep. So I don't know if anybody gets 8 hours of sleep anymore, but certainly it is important to get a decent amount of good quality sleep, because that contributes to our protection against Alzheimer's disease. If we're helping to clear the trash out of the brain every night, when that clearance can be at its optimal function, then that's going to help us. If we're stressed out, if we're getting poor sleep, then that's going to be detrimental to us. And I just mentioned something that I'm reminded myself. If you look at these features, I know I'm circling back on a theme I already mentioned. Nothing operates in a vacuum here. So many of these things are linked together. Why might you have disrupted sleep? Well, you might have disrupted sleep because you're stressed about something, or because you had a head injury. Folks with head injury have, very well documented poor sleep, disrupted sleep, sleep apnea, obstructive sleep disorders. The physical activity, the mental activity, the diet, these other features, they all work together. our diet, improving our diet, that can boil down to not eating too much pro-inflammatory material, anything that might cause pro-inflammatory responses. So if we're eating a good diet that's rich in antioxidants and low in inflammatory compounds, that's going to help. So all of these things are all linked together. So I'm going to focus on those three at the bottom there: physical, mental activity, and diet, because these are three things that we can all do something about. And I think that's really good news. Other things we may not have so much control over, like stress, but physical activity and mental activity and diet, we can certainly do something about. Now, again, we've known for years, there's been these concepts around forever, the use it or lose it idea. And, about brain activity. And again, we can all guess that a healthy diet is going to be better for us in lots of reasons, not just for Alzheimer's disease. The difference now is that there actually have been properly designed scientific studies, randomized controlled trials, where folks have investigated the effects of these variables on risk for Alzheimer's disease. So the FINGER study, which is a Finnish study, a large study in the Finnish population, where they had an intervention of physical activity, mental activity, and diet versus just normal counseling. And the MIND study, where they specifically looked at a Mediterranean diet, a Mediterranean type diet, which was a largely pescatarian type diet with fruit and vegetables as well. So these studies have now been done and are showing that we do have reduced risk for Alzheimer's disease if we engage in these activities. So yeah, what can we do to avoid these changes? The three things I just mentioned. The first thing really is to control our existing conditions. We can't do much about our medical history, but if, for instance, we have high cholesterol or high blood pressure or diabetes, we have treatments for those these days. We have ways of managing those. We should manage those because they all contribute risk for Alzheimer's disease. Then separately from that, it's lifestyle interventions. So here's these are some pieces of data which I think are just nice to see, because it's quite remarkable to see how much control we have over our own bodies and over our own destiny. This is looking at the hippocampal volumes, which you wouldn't really think that you could do a lot over the years to affect your hippocampal volumes, but actually it turns out you can. And as I said, the hippocampus, that's where the new information comes into the hippocampus to begin with, and then it sends the information out to the cortices. So if the hippocampus is fit and healthy and buffed up, it's going to do a better job. But just with age, unfortunately, like so many other things, the hippocampus starts to, wind down a bit. And so there were studies done to show that physical activity can increase hippocampal blood flow and hippocampal size and improve spatial memory. So we can actually change the size of our hippocampi with physical activity. And this study, the data on the right there, looked at adults aged 62 to 79. I think there were about 90 people in the study. So this isn't young individuals. This is older adults who were starting to show shrinkage of their hippocampi. And over 12 months, the ones doing a high level of physical activity, shown on the left here, had a much less reduction in their hippocampal volume versus the ones engaging in low levels of physical activity. Like it's just a few percent change in the volume, but that's only over a year. And this is in older individuals. So you can do things to maintain your hippocampal volume and its degree of activity. I love this story. This is an old one, 10 years old. This is before the days of Uber and Google Maps. Back in the day when taxi drivers used to have to memorize the A to Z. Do you remember the A to Z? You used to get these. I know when I went to London, I got an A to Z. It was like the Bible, and that's how I found my way around. And I'm sure there's one in New York and many other cities as well. So the London taxi drivers used to have to go through this 4 years of training where they learnt the A to Z, so that anybody could jump into a cab in London, give any sort of obscure address, and the taxi driver would know where it was. Remarkable. So 4 years of training. And if they got what was called the knowledge, then they were qualified. And the graph here shows the volumes of the hippocampi in the individuals at the start, shown in blue, and at the end, shown in orange. And you can see the controls on the right, there was no change. The folks who did the training and didn't qualify, they also showed no change. But the folks who qualified, they've shown a significant increase in the volume of the hippocampi. So again, just a real example of the use it or lose it concept. You keep your mind active, keep your brain active, and you can really buff up a reserve against whatever the future might hold. Now this one I do like. Well, I like the others too, but this one's particularly nice if you don't always want to adhere to a really, really strict diet. So this is looking at the risk of conversion from mild cognitive impairment to Alzheimer's disease in relation to diet. And again, it's looking at the Mediterranean diet. So on the left, taken at 100% risk for conversion, is the folks on the, what we call the Western diet, the burgers and fries and hot dogs and sodas and everything packed with sugar. If you have a medium adherence to a Mediterranean diet, and I'm sorry, I forget exactly what the definition was of medium adherence, but you can see that you cut your risk by nearly 50%. This is folks who already have mild cognitive impairment, but their risk of progression was reduced by 50%. And the really good news is that even high adherence didn't change things that much more. So to me, this says, so long as you're doing, reasonably okay with your diet, you can still have a bag of cheese puffs once in a blue moon, or, an ice cream or a big dinner down at, wherever. It's encouraging, I think. And then this is another study looking at both physical exercise and diet together. So again, the greatest risk for Alzheimer's disease, low levels of activity and a really poor diet. But then if you change one of those, either if you improve your diet or increase your amount of activity, you cut your risk quite considerably. And then of course, best of all, high level of activity, high adherence to a good diet really reduces your risk. As I say, these are studies now, and they're starting to come out because these studies, of course, take time. They're conducted over years. They're prospective studies watching whether folks convert to Alzheimer's disease or not. But we now have real data to back up all the anecdotal stories we've had over the years. Now real data to support the fact that these lifestyle changes can have an impact on our risk for Alzheimer's. So that's all about protecting against getting it, prevention, or at least pushing it off till as late as possible. So what about actual treatment when folks actually have the disease? So as Lucy mentioned, my introduction into all this was back during my PhD. I was working on genetic analysis of families with early onset Alzheimer's disease. So these are very rare families which get Alzheimer's disease sometimes as early as their early 30s up to their early 50s. And we were studying families like the one on screen here, where every shaded in individual, this is the grandparents and then the parent generation and the children. All the shaded symbols would be folks who developed Alzheimer's disease. And what we were looking at was to try and see within a single family, and with lots and lots of families, but within individual families, was there anything in common? If we looked across their entire genome, all of their genetic information, what was in common that was being inherited from each Alzheimer person to the next? And summarizing a lot of research very quickly, that led us to the identification of genetic errors, actual mutations in the amyloid gene, the gene that encodes that amyloid protein I showed you right at the start. So if you actually have mutations in that amyloid gene, we showed that gave you Alzheimer's disease with pretty much 100% certainty. Now, as I mentioned, these amyloid mutations are very rare. And the mutations, these early onset mutations that cause Alzheimer's disease account for less than 5% of all Alzheimer cases. But it did give us clues as to disease mechanism and ways that we might be able to intervene. And it also enabled us to create animal models. So by identifying mutations that could cause the disease in humans, we now had a way to model Alzheimer's disease in the lab. So whenever we introduced these genetic errors into mice, it turns out the mice were born normally and developed normally, but as they aged, they developed cognitive problems and developed amyloid plaques. So now we have a tool, a platform that we can investigate the disease progression in those mice, and we can also test any treatments that we think might have some value. And the reason you're seeing a mouse swimming around there is people always ask me, how do you know if a mouse has Alzheimer's disease? So we have behavioral tests, one of which is called the Morris Water Maze, which is where we put the mice into a pool, kind of the size of a kid's paddling pool. And there are visual cues around the outside, like the angelfish you see there. And there's a submerged platform. And mice, unlike rats, don't like to swim. So they actually like to find the platform, but they can't see it because they're swimming with their head up, but they can see the visual cues. And so a normal mouse, if you put him in the pool several times in a row, he will learn that the platform is near where that picture of the fish is, but the Alzheimer mouse will not learn. And so that's one of the tests that we do for cognitive function in the Alzheimer mice. So having identified that changes in how amyloid is being produced, specifically too much of it being produced, could cause Alzheimer's disease, this immediately presents us with some ideas of how to treat Alzheimer's disease. So this cartoon is a representation where the brain is the basin here and the neurons here inside. So we could either shut off the faucet, shut off the supply of amyloid. So if we can inhibit amyloid production, that would be that approach. We could increase the clearance of amyloid out of the brain, or we could tackle whatever it is that amyloid does to neurons that is so bad. So those are kind of the three big picture approaches to targeting amyloid in Alzheimer's disease. So summarizing, jumping 10 years or so, with a lot of assays in our labs to try and identify compounds that could have these effects, we identified a drug called nilvadipine. Now this is a drug that was already in existence. It's an antihypertensive drug that had been used for many years in Europe and Japan. And all of our data showed that it worked in our mouse models of Alzheimer's disease. Now I've just got a couple of pieces of data for you here. This looks at the amount of blood flow in the mice. So the mice, these are all Alzheimer mice, and we're scanning the brains of the mice to see the level of blood flow. And the more red there is, the better the blood flow is. Just like in human patients, where we know that they get reduced blood flow. On the left are mice that were treated with placebo, and on the right are mice that were treated with our compound, nilvadipine. And I hope you can see that there's a very significant increase in the mice that were treated with nilvadipine versus the untreated Alzheimer mice. So big effect on blood flow. We also showed improvement in cognitive function. We also showed improved clearance of amyloid out of the brain into the blood. And then this is a picture showing sections, brain sections from the mice. And the top three are sections from placebo-treated mice. All of those little black patches you can see there, those are amyloid plaques in the brains of the mice. And I hope you can see in the bottom three pictures that nilvadipine treatment significantly reduced the amount of plaque in the mice. Now, importantly, it's worth mentioning, we didn't begin treatment until the mice were 10 months old, at which point they already had cognitive deficits and they already had amyloid plaques. So we were trying to mimic giving the treatment to a patient with Alzheimer's disease, not giving it to them 20 years ahead of any clinical presentation. We were mimicking active treatment of Alzheimer's disease. And nilvadipine looked very promising. And I will come back to that in a moment. But while we were doing all of this, the drug companies were focused largely on an antibody approach to Alzheimer's disease, because it had been shown that in mouse models, the mice carrying the mutations that my colleagues and I found in London, in those Alzheimer mice, when they were vaccinated, if you will, with amyloid, it actually reduced the amount of amyloid in their brains. So the first trial, and it kind of went through rather hastily. I just realized that's been mimicked recently, but anyway, went through a bit hastily. And there was a trial where folks were actually being administered amyloid as a treatment. And that trial was halted because several people died. And when the investigations were carried out, they had an awful lot of brain swelling. And it seemed like that might have been related to a sudden rush of clearance of amyloid out of the brain, basically causing hemorrhages and all sorts of problems. So the approach was then modified, rather than to sort of the vaccination approach, to actually do approaches with antibodies to amyloid. So the idea there being that the amyloid would get drawn out of the brain and caught up, bound by the antibodies, and then excreted. And as I'm sure many of you are aware, there have been very many trials of anti-amyloid antibodies in Alzheimer patients. They have been designed against different forms of amyloid. They've been designed against soluble amyloid. They've been designed against aggregated amyloid, different fractions, different fragments of amyloid. By and large, they've all shown the same data as I'm showing here, which was from the Eli Lilly drug, bapineuzumab. And what you're looking at here is their performance over 78 weeks of an Alzheimer clinical trial. Quick point about that, Alzheimer's is a really expensive condition to work on, because apart from anything else, the clinical trials that will lead to, hopefully, approval of a drug need to last for 78 weeks, because you need to see sufficient decline in the placebo group in order to see a significant effect in the treated group, if there is going to be one. So they typically last for 1.5 years. And we're looking at the ADAS-Cog, which is the Alzheimer's Disease Assessment Scale, and the decline in that, which is basically people getting worse. And I'm afraid here you can see in black is the placebo group declining over the 1.5 years. And unfortunately, the treated folks in blue declined in exactly the same way. So no positive signal from this study, and there have been many, many others like it. This brings us to the question that I touched on earlier, targeting amyloid, even targeting tau. Is it too late by the time somebody has established dementia? Is it too late to just go after amyloid and tau? You've already got all these other things going on in the brain. And do we need to be thinking about something that tackles multiple different things, like the drug we were looking at? It reduced amyloid, it reduced tau, it also reduced inflammation. And there's been a lot of discussion about the fact that Alzheimer's treatment is going to require a multimodal approach where multiple things are being targeted. But the poor data from the clinical trials with anti-amyloid approaches have led to the clinical trials recruiting earlier and earlier stage individuals. So more recently, trials have included folks really only in the very early stages of Alzheimer's, or folks diagnosed with mild cognitive impairment, in an attempt to try and get in early. Now, aducanumab, Lucy asked me to comment on this. So why is there so much controversy about aducanumab, which it just got rebranded as Aduhelm? It has just been approved by the FDA. I will hopefully explain a little bit of the controversy. So aducanumab is an antibody against amyloid, which was developed by Biogen, and they had some positive results in a phase 2 trial that encouraged them to move on to 2 large phase 3 trials, and the data are shown here. They were called Emerge and Engage, and each had about 1,500 individuals divided into roughly 500 placebo, 500 low dose, 500 high dose of the antibody. Now, let me also be clear, these antibody trials are not easy. These are infusions. You have to go to a clinic. The dosing was, I think, once a month or once every 2 weeks, depending on which regime, and not cheap either. I'll come to that in a moment. In these plots, we're looking at the change from baseline in the clinical dementia rating scale, the CDR. So this time, the more upward the trajectory, the worse people are doing. So here in gray, you can see the placebo effect in both Emerge and Engage. And in the Emerge trial, you can see in pink that the folks who were in the high dose of aducanumab did show a significant difference by the end of the 1.5 year study. However, the Engage trial did not show a difference. So this is the first problem. The FDA, up until this point, required 2 positive phase 3 trials in order to approve a drug. So that's point number 1. Biogen actually announced cessation of this trial in March of 2019 because they did a futility analysis, which was all factored into the plans for the study, where they took a look at the data they had at that point and predicted whether or not they were likely to get a positive effect. And they determined they were not likely to. And so they stopped the trial in March of 2019. But in October of 2019, they had reanalyzed the data, looked at it a few different ways, carved it up a few different ways, and they went back and said, no, actually, we got a positive signal with eMERGE, and we want to come back. And that's part of the reason for the controversy. The trial design was rather messed up. They had 4 amendments, I think, and there was confusion over how many people were getting the high dose, and some people switched from low dose to high dose. And at the end of the day, as I've said, only one of the trials showed any possible positive. The other thing is that if we look at the adverse events in this trial, and I know there's a lot of data on here, but just focus here, ARIA-E and ARIA-H. If you look at the percentage of individuals who had adverse effects here, this is in the treatment groups. There's not much difference between placebo and treated for these other features like headache, 15% versus 19%, but ARIA-E was 25 and 34 versus 2, and ARIA-H 13 and 9% versus 2. ARIA is amyloid-related imaging abnormalities. So these are problems you can pick up with MRI imaging. And the E stands for edema, so brain swelling in these individuals, significant brain swelling, which meant that they stopped getting treatment for a while. These symptoms did resolve. ARIA-H is hemorrhaging. Again, these did resolve in time, but folks either stopped treatment or dropped out of the study, or they continued at a lower dose. They dropped back to a lower dose. So there's not insignificant adverse events associated with this treatment. And of course, people actually in the trial were getting very frequent MRI imaging in order to monitor this. So what's going to happen now that it's been approved? So this is why there's lots of issues, and I can talk more about this if anybody wants to, but this is why there's been a lot of controversy. The scientists said it should not be approved. The FDA was under enormous pressure from advocacy groups and everybody else. And they did approve it, but they have approved it on condition that Biogen conducts another phase 3 trial. And they have to have that completed within 9 years. And obviously that has to be positive. So they kind of found a way through, the FDA. They've let this go through because they know there's such a demand for things to move into clinical trial for Alzheimer's, into approval for Alzheimer's. But they've made it clear they don't think the data are sufficient at the moment. So that's why there's been so much controversy. Now add to that the fact that this drug is going to cost about $56,000 a year. And at least one insurance company that I know of has said they will not pay for that because it's an investigational drug still. So we're in limbo at the moment as regards what's going to happen with aducanumab. So very quickly, because I see the time is ticking on, I'm so sorry. I just want to show you back on the Roskamp work, just with our little group, as opposed to Big Pharma and all of their millions. This is what we've done with our lead drug I showed you, where we saw all the positive effects in the mouse model. We did an early study, just a small study to show that it was safe to give it to Alzheimer patients. We saw no ill effects. We have identified at the molecular level how the drug is working. And we were able to do a phase 3 trial in Europe, not as big as the big pharma trials. This is 500 patients, but it was funded by the European Union. And we managed to do it for about $10 million, which is about, it's a very small fraction of what those other trials cost. So our data, or our trial rather, did everything as if the trial would have been done here. We modeled everything the way it would normally be done, 18 months of treatment. This is a pill that people took in the morning, a slow release tablet, just one a day, and we had all the usual outcome measures. And we had about 250 patients in each arm. Now, unfortunately, our recruitment, we were not able to focus just on folks in the very early stage of the disease. So we had some folks who were quite severely demented at the time they began the trial, and you'll see in a minute why that's important. So overall, at the end of our study, we did not see any significant difference between treated and untreated individuals. However, when we looked, and these were analyses that we planned before we even started the trial, when we looked at the data, we saw that there was a difference depending on where people started from. In other words, what stage of the disease they were at whenever they first started treatment. And so because we had already planned to do that, and because we saw the difference, that statistically allowed us to go back and reanalyze the data and just focus on folks in the early stage, who were in the early stage of the disease when we started treatment. And these are the results we got. So this time we're looking at the mini-mental state examination, which for those who are familiar, it's a 30-point scale. So if you score 30, you're cognitively normal. Folks who score lower, if they get to 25, you would definitely know there were cognitive issues. And there were folks in our trial going down as low as 13, 12 on the scale. So we had some very severely demented individuals. But if we just looked at folks who began the trial with a mini-mental state examination score of 25 or above, you can see this is the placebo line, dotted line is the placebo line, that's their decline over 78 weeks. But in our treated group, they didn't decline anywhere near as much. And by the end of a year and a half, we got a statistically significant difference between the two groups of 47%. So in other words, we slowed the decline by 47% with our treatment. And now we get to the business part of what I do. So pharma companies were very interested. We don't have the funding to move forward. That's why we spin out these for-profit companies to try and do these clinical trials, because the Institute, the basic research of the not-for-profit Roskamp Institute is entirely supported by the money we bring in on grants from the NIH, the Department of Defense, and the VA. So we cannot fund big clinical trials. So we go to big pharma with these data, and they were very excited about the novel mechanism of action and the novel molecular target, but they weren't interested in nilvadipine, because our patent on nilvadipine, we'd been working on it for a long time at this point, so we only had a few years left. So they would never have been able to recoup their money because it would have gone off patent in the next few years. Moreover, nilvadipine then became unavailable in Europe and Japan to treat hypertension. So we weren't even able to carry on any other studies academically funded. So what we did was we went to the National Institutes of Health over here and presented our research, and we are now funded by the NIH to develop new compounds, more potent compounds, we hope, based on the nilvadipine chemical structure and the nilvadipine mechanism of action. And those new compounds will be new chemical entities. So in other words, from the business side again, we will be able to get new patents on those drugs, and the pharmaceutical companies will be interested at that point, and we will be able to move forward with those into clinical trials. So I'm just finishing with one slide. I mentioned toxins very briefly early on, and Lucy mentioned the Red Tide project. How does this link into anything I've just been talking about? Well, in 2018, when we had the appalling bloom here, which sadly I think is about to be repeated this year, we saw the fish in the bay, we saw the manatees, turtles, dolphins on the beaches. Our neurologist friends in our clinic and elsewhere around the Sarasota and Manatee area were telling us that they had an increased number of patients coming to them saying that their neurological signs and symptoms were worse, and they were sure it was because of red tide. And so this obviously got us very interested. Then we see that a lot of the features that we saw in manatees and in dolphins have parallels with what happens in neurodegeneration, cognitive decline. And it's kind of an obvious question. If we've got a neurotoxin out there in the Carina brevis, in the brevetoxin, that is a neurotoxin that is present, if it's present in enough quantity to kill manatees and dolphins, what might it be doing to human health? And it really had not been studied. Nobody had really studied this. I mean, people knew that they got sick. Humans would get sick if they ate shellfish that had been exposed to Carina brevis, but nobody had really looked to see what about other features, what about other neurological signs and symptoms. So we launched our Red Tide project, and we self-funded it to begin with to get it off the ground. And we generated preliminary data, and we submitted it to the NIH, and we got funded. And so we have an ongoing study where we're recruiting folks in the area. We're getting blood samples, and we're asking questions about any neurological signs and symptoms. And the idea is to collect data and blood samples from people during a period of no bloom and during a period of a bloom. So as you can guess, we're pretty busy at the moment, but we're still recruiting. We had a goal of 250 individuals. We reached that goal. We've now raised the bar. We now want to get 400 individuals. And I can tell you that we are able to detect brevetoxin in the samples we're collecting, which means that the neurotoxin from the red tide algae is getting into the bodies of folks here in the Bay Area. So I will leave you with that. I hope that the positive note from the Alzheimer's side is that we have trials ongoing, we have drugs coming down the pipeline, and there's a lot we can do in the meantime to help protect ourselves against getting Alzheimer's disease. So I'll stop there, and I'm sorry I talked for so long.
Well, thank you so much. That was so incredibly useful and, you know, give us so much information. We have lots and lots of questions. I don't know how many we'll get to them, but let's start with Beth Gottfield. You want to ask your question? Unmute yourself.
So yes, I had a few. First of all, for this Red Tide project, if you can put in the chat, you know, how we can sign up for it, that would be helpful, or David Patterson's contact information.
Absolutely. I'm going to have to open up my screen again a second just to remind myself. Hang on.
Okay.
Okay. I will do that. I'll put it in the chat.
But in a lot of our questions that we've asked, I see were answered. But one question I wondered is, are we seeing more Alzheimer's now? I mean, I've had 2 deaths this week of Alzheimer's, for example, on my block. But I just wonder, are we seeing it more than we did decades ago because of whatever environmental influences, all the things you said, or not really? It's really because we're living longer, or of course.
A really good question. So very interestingly, we are seeing more, and there have been projections that it was about 4 million Alzheimer's patients in the US in about 2010, I think, and projections that was going to reach like 15 million by 2030, something like that. And a lot of that is about, and worldwide, the incidence is increasing because, of course, folks in less well-developed countries are living longer, increased healthcare, we're surviving other things. So we're kind of living long enough to get Alzheimer's disease. But I did actually read a review just a day or so ago that suggested that it might be tapering off a little bit, that there might be a little bit of a reduction, possibly because of folks reducing their risk factors, folks tackling, you know, things that have been risk factors like smoking, which of course is a big risk factor for vascular disease. So overall, the risk for vascular disease being decreased will help with risk for dementia. So in general, I think it is still increasing. And of course, you know, it does increase with age. And we have an aging population. Well, everywhere has an aging population, but here in Sarasota, of course, we have really healthily aging individuals. So we've got folks around who are still, you know, marvelous, you know, right up to whatever age. And I think it is still increasing, maybe not quite as horrifically as it was a while back.
Thanks. My other one was on genetics, and you answered it, so thank you.
Okay.
Mindy, would you like to ask your question?
Sure, and you answered questions 2 and 3, but question 1 was regarding Paul Cox's research on L-serine. A couple of years ago in Fortune, there was a really in-depth article about the research institute he's got, and he was studying bats in one of the small island nations and a toxin that the people were ingesting, and then a group of people who previously did not have any history of dementia or Alzheimer's were fast developing it, and his research is focused on L-serine. I don't know if you have any familiarity with that, but I am looking for something preventative to do on top of everything else you suggested, because I have a lot of family history of dementia and Alzheimer's, and I don't want to get it.
Very understandably, I'm not familiar with that work. The toxic exposures are more traditionally associated with Parkinson's. We know that Parkinson's dementia and Parkinson's-like symptoms can come from environmental exposures. But again, just speaking to the fact of everything being linked together, the reason that we also work on Gulf War illness, we were attracted to that because of the cognitive presentation in folks with Gulf War illness. These are folks who were exposed to massive doses of pesticides, also prophylactics against nerve gas attack, and those are the two main things that seem to have contributed to Gulf War illness, which has multiple symptoms, but one is cognitive loss and problems there. Interestingly, if we look at other sort of environmental toxic exposures like pesticides, things like that, there have been big studies done, for instance, with farmers in Utah. There was a big study a few years back showing that farmers exposed to organophosphates had increased risk of Alzheimer's disease versus those not exposed. And so what I can tell you that what we see in the brains of individuals with Gulf War illness and in our mouse models of Gulf War illness, we see interesting and quite distinct patterns of inflammatory and immune activations. So again, these things all play together. And as soon as you have some of these problems going on in the brain, you're presenting a compromised environment. So if a subsequent insult comes along, you're less well able to take care of it. So we don't have the specifics yet of how these particular toxins bring about these changes. But yeah, the bat story sounds very interesting. I mean, I'm reminded from that, it ties in also with red tide, of course, but also there was a big study years ago in Guam where there was the whole population developed ALS, and it was something to do with the a particular toxin that was specific to Guam. I don't think it might have been a blue-green algae there, but I'm not sure. But again, that was an environmental toxin.
Thank
There
you.
are several questions that had to do with the new drug, which I think you've answered. So Karen, would you like to ask your question? Unmute.
Yeah, good. Thank you. First of all, let me just say thank you, Fiona, for a wonderful, wonderful talk. And I want to say how lucky we are to have the Roskamp Institute in our community. This is just such an asset. I just asked a very simple question: is tau present in all neurons? And if so, does it have any effect in other neurons outside of the brain?
It does. It's present. It certainly has a role in the spinal cord. I know that because I know whenever we fiddle around with tau, fiddle around being a very scientific term, when we fiddle around with tau, we can see changes in spinal cord pathology. I should know a much better answer to that, Karen. That's a really great question. But yeah, in the brain, it's everywhere. It's part. It's a microtubule associated protein.
Right. Oh,
it's
interesting.
in all of them. Yeah.
Terrific. Thanks.
Monica, you had a question that sort of was on many people's minds.
Yes, thank you. Thank you. Thank you for the talk, Fiona. I really got to understand very well the disease, and your clarity was great. Like my father has Alzheimer's, and that's why I'm, getting all the information I can. And I was wondering, like, in your experience, what is the ideal treatment for the family in terms of getting to know how to deal with the person, and how to deal ourselves with our emotions and with all these, desperate feelings that we have that we are losing him, and that, that everything is changing. So I just want you to ask you that. I know you don't live in Mexico, but of course I can, find groups here, but I don't know what is the ideal treatment for families. What is your recommendation on that? Thank you. And thank you to all.
Thank you. Oh, such an important question. I don't know if there's an ideal treatment, but I do know that the stress on the families is enormous. And I think if you can find a way to help you deal with your stress, whatever that may be, if it's doing something together. I mean, I don't, I'm very fortunate. I don't have that stress. What I do to deal with my stress is yoga and meditation. That works for me, but it doesn't work for everybody. So I think as a family, able to work together with that, I think that in and of itself, I think if you're all together, understanding the experience, I mean, you've got your own caregiver support group there almost, and if everybody's going through the same thing, patience and understanding that you're, not fighting the patient. I see a lot of people sort of arguing back, the patient will be saying something, and instead of going with it, if the patient is saying that they had breakfast already and they didn't, it's like, okay, so you didn't, well, now this is lunch, just play along almost, and you unfortunately do have to accept it's almost a different person, your father as he was, as you knew him, he's moving on at the minute. You're now going back. He's going to be going back through his life at some point, if not already. He'll probably start to recognize his children as his siblings. That's a very common thing. And that's devastating for the children. It'll be devastating for your mother in that situation. So knowing what to expect, I guess, is also important, so these things don't catch you unawares. But having the support of your family and with you all, getting an awareness of what to expect and knowing what's coming. I mean, at the moment, sadly, what's coming, and so you can hopefully find a way to face that together.
I think, Natalie, you had several questions. Some I think may have been answered, but I think there are a few that weren't.
Thanks. I had a couple. The first question I think you really touched on very well, which is potential prevention. And I'm actually very interested in looking at the finger study and the mind study. So thank you for that. The second question I have is really more about a much newer technology called the CRISPR technology, which is around gene editing. And are you familiar at all with any work that's being done or investigation work that's being done, possibly using that within Alzheimer's, either to prevent or treat?
Well, we use it all the time in the lab, in our cell models. Mm-hmm. But as regards using it as a therapeutic is another massive step, because of course, in that instance, you've got thousands of millions of cells that you would need to edit. Mm-hmm. So I think the main issue at that point, and it is something that folks are looking at for sure, the main issue with that is about delivery and how do we get that very specific delivery to this particular cells we want. Delivery is always a concern. Like, forget CRISPR, it's a concern with everything. I mean, so many drugs that we've looked at look as if they would be absolutely fantastic, except they don't get across the blood-brain barrier. So they, you pop them in your mouth and they could go everywhere in the body and they can't get across that protective, barrier into the brain where the action needs to be. So, these are all things you learn as you go down the path. So delivery to the cell types of relevance is, I think, the main concern with CRISPR. And of course, folks have concerns about, about genetic technology in that regard, just like they do about the mRNA vaccines, right? It's like, oh my gosh, what's this going to do to me 5 years from now? Am I going to be doing something crazy, growing another head or something? I don't think there's any reason to be concerned like that, but it's still a very early technology from the therapeutic point of view. As a research tool, it's fantastic. And just for anybody who doesn't know, it's a way of going into the genome of a cell or an animal and just very specifically editing the genome. And so you could take all of the neurons in your petri dish and totally change out the gene that's there. And an example that we have is, we might replace, if we're dealing with normal healthy neurons, we might edit those so that they overproduce a protein or underproduce a protein or produce a protein they never normally make, and see what happens. What happens if astrocytes start to produce tau, for instance, things like that.
Did I miss anyone's question that they want to ask? If you do, just unmute yourself. All right. Well, thank Fiona. This was fantastic. Thank you so much for all the information and for everyone. There was a lot of good suggestions that people gave of caregivers and places you can get help. So just look, hopefully you saw the chat and were able to take that information down. So thank you all for coming and joining us. And we're having actually in August, on August 19th, we have a luncheon, bring your own Zoom, and Carol H. Green is going to do it, and she's going to lead a conversation talking about sort of decocooning from COVID, or recocooning, since we don't know exactly what we're doing. So it's at noon. Hope you'll join. It's just informal conversation. So thank you again, and take care, everyone. Stay well.
Thank you. Thank you.
Thank you. Give us 1 second
Thank you.
so that we can get the information on the red tide study.
I did put it up there. I can't see it now. I did put it in the chat.
It's in the chat,
Oh,
but
I
we
missed
can
it.
we can ask Tanya to send it out.
Okay,
Will
great.
will the Zoom recording
there it
be
is.
available?
Yes, I believe so. I'm sure it's on our website. I'll check with Tanya as to how it is, and we can send all of you who registered a note about it. I know I need to look back at some of the science. As I told Fiona, I didn't take science in school, so this is above me, way above my pay grade, but it was interesting. All right, thank you all.
Absolutely.
Take care. Thank you so much, Fiona.
Thank
We
you.
really appreciated it.
That's great.